Today, we will talk about gene editing and personalized medicine. Personalized or precision medicine combines genetic information with phenotypic and environmental characteristics to produce healthcare tailored to the individual and eliminates the constraints of one-size-fits-all therapy. 1. TRISPRCAS9 TRISPRCAS9 is a revolutionary gene editing tool derived from a bacterial defense system. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which, alongside the Cas9 protein, allows precise modification of DNA in organisms. By targeting specific genetic sequences, CRISPR-Cas9 can add, remove, or replace DNA, offering potential solutions for genetic disorders. Its efficiency, accuracy, and relative simplicity have made it a preferred choice for genetic research, promising advancements in medicine, agriculture, and bioengineering. However, ethical concerns arise, especially with germline editing, which affects future generations. Still, CRISPR-Cas9 remains a transformative technology in modern science. 2. Zinc finger nucleases. Zinc finger nucleases, ZFNs, are synthetic proteins designed to target and manipulate specific sequences within a genome. They function by combining a zinc finger protein, which recognizes and binds to a specific DNA sequence, with a foci nuclease domain, which induces DNA breaks. Once the DNA is cut, the cell's natural repair mechanisms activate, leading to the addition or deletion of genetic material. ZFNs paved the way for targeted gene editing, offering potential therapeutic applications for genetic disorders. While revolutionary in their time, the emergence of newer gene editing tools like CRISPR-Cas9 has overshadowed ZFNs due to their simplicity and efficiency. However, ZFNs remain significant in the history of genome engineering. 3. TALENs. Transcription activator-like effector nucleases, TALENs, are engineered proteins designed for targeted gene editing. TALENs function by combining a customizable DNA binding domain, derived from TALE proteins found in certain plant pathogenic bacteria, with a foci nuclease domain to induce DNA breaks. Like other gene editing tools, once the DNA is cleaved, the cell's natural repair mechanisms can be harnessed to introduce specific genetic modifications. TALENs offer greater precision in targeting specific DNA sequences compared to earlier genome editing techniques. 4. CAR-T cell therapy. CAR-T cell therapy is an innovative form of immunotherapy in which a patient's T cells, a type of immune cell, are genetically modified to produce chimeric antigen receptors, CARs, on their surface. These CARs enable the T cells to recognize and attack cancer cells more effectively. The process involves extracting T cells from a patient, genetically engineering them in a lab to express the CARs that target specific proteins on cancer cells, multiplying these modified cells, and then reintroducing them into the patient's body. CAR-T cell therapy has shown remarkable results in treating certain blood cancers, especially those resistant to other treatments, marking a significant breakthrough in cancer therapeutics. 5. Genome sequencing. Genome sequencing refers to the process of determining the complete DNA sequence of an organism's genome. This comprehensive mapping of all the genes in an organism provides a detailed genetic blueprint, offering insights into its physiology, evolution, and potential susceptibilities to diseases. In humans, genome sequencing has paved the way for personalized medicine, enabling tailored medical treatments based on an individual's genetic makeup. Technological advances, such as next-generation sequencing, NGS, have made sequencing faster and more affordable. As a result, genome sequencing has expanded from research environments to clinical settings, providing deeper insights into genetic disorders, ancestral heritage, and guiding therapeutic interventions. 6. Pharmacogenomics. Pharmacogenomics is the study of how genes influence an individual's response to drugs. This field combines pharmacology, the study of drugs, and genomics, the study of genes and their functions, to develop effective and safe medications tailored to an individual's genetic makeup. By understanding genetic variations that affect drug metabolism and action, pharmacogenomics seeks to reduce adverse drug reactions and ensure that patients receive the most appropriate treatments. The ultimate goal is personalized medicine, where healthcare providers can select treatments based on a patient's genetic profile, ensuring optimal therapeutic outcomes with minimal side effects. 7. Liquid biopsy. Liquid biopsy is a non-invasive diagnostic method that detects and analyzes circulating tumor DNA, circulating tumor cells, CTCs, and other molecular biomarkers from bodily fluids, primarily blood. Unlike traditional tissue biopsies, which require surgical extraction of tumor samples, liquid biopsies offer a less invasive means to obtain genetic information about tumors. They are particularly valuable for monitoring disease progression, treatment response, and detecting minimal residual disease or potential relapses. Liquid biopsies hold the promise for early cancer detection, tracking tumor evolution, and guiding personalized therapeutic strategies based on the molecular signature of the tumor obtained from the patient's blood sample. 8. Polygenic risk scores. Polygenic risk scores, PRS, are a computational tool used to estimate an individual's genetic predisposition to certain diseases or traits. PRS is calculated by aggregating the small risk contributions from thousands of genetic variants identified in genome-wide association studies, GWAS. By tallying up these risk variants, PRS provides a cumulative score that helps predict the likelihood of developing specific conditions, like coronary artery disease, diabetes, or certain cancers. While PRS offers valuable insights, its predictive power varies across diseases and populations. 9. N-of-1 trials. N-of-1 trials, also known as single-patient trials, are clinical studies in which interventions are tested on a single patient, often using crossover designs where the patient receives both the treatment and placebo at different times. These trials aim to determine the optimal or best intervention for that specific individual. N-of-1 trials can be especially valuable in personalized medicine, where treatments are tailored to individual needs. By comparing the effects of different treatments or treatment combinations within the same patient, clinicians can make more informed decisions about the most effective and safest therapy for that particular person, thereby optimizing therapeutic outcomes. 10. Individualized drug compounding. Individualized drug compounding refers to the tailored preparation of medications to meet specific patient needs. This practice allows pharmacists to create custom drug formulations when commercial drug products do not adequately address those needs. For example, if a patient is allergic to an ingredient in a commercially available drug, a compounded medication may be created without that allergen. Other reasons for compounding include altering drug dosages, creating alternative drug forms, like liquid versions of solid medications, or combining multiple medications into a single dose. What do you think of our video? Let me know in the comment section below. Before you go, please hit the like button and subscribe to my channel. Thanks for watching.